Welcome back, everyone. Dr. Hall here, and we're going to talk about the first step in an action potential, which is actually reaching threshold and deciding to send an action potential. So if we look at the outline for lecture 3, you'll see that as I got into it, I expanded and divided it up. So we're going to do 3B now, which is just talking about EPSPs, IPSPs, and reaching threshold, and we'll do the other aspects of the action potential in separate chunks. All right, so let's talk about how we reach threshold. So remember the axon hillock, we talked about that when we talked about axons and how it's the part of the neuron that decides whether or not to send a signal, right? So it's constantly getting information, some saying you should send an action potential, some saying you shouldn't, you should, you shouldn't, you should, you shouldn't. So how does it actually decide what it's supposed to do? When is it supposed to send an action potential and when is it not? So all of those signals that are coming in from other neurons. And anytime we have a neuron connecting and communicating with another cell, we call that a synapse. So we have on our neurons tens to sometimes 50 or even more synapses. So places where you're getting incoming information from other neurons. Now, for the most part, this is happening, of course, on the dendrites, many of which have these spines, right? These little spines where you can receive information. But neurons can also have synapses and receive information on their cell body or soma. So they also have synapses there. The neuron that is receiving these signals is called the postsynaptic neuron because it's after the synapse, right? It's on the back side after the signal has been received. And those signals are going to cause small changes in voltage across the cell membrane, across the plasma membrane, which are called postsynaptic potentials or PSPs. So we're going to talk a lot about those. So let's kind of get ourselves situated. Oh, I forgot, there's an animation, sorry. So here's all my little signals coming in, and they're going to be changing voltage and causing these postsynaptic potentials. All right, sorry about that. Okay, so what is happening at those synapses is that the presynaptic neuron is releasing neurotransmitters. We haven't talked about neurotransmitters yet because we haven't really talked about synapses in depth yet. We will get to that soon, next week actually. But what happens is the neurotransmitter binds to a type of ion channel called a ligand-gated ion channel, which you probably remember is the type of ion channel that will open when a certain molecule binds to it. So those neurotransmitter molecules being released by a presynaptic neuron, the neuron that's communicating to our neuron, releases neurotransmitter, which binds to a ligand-gated ion channel, which then opens. And then depending on which ion it is specific for, and depending on the electrochemical gradient of that ion, those ions will move. So when this happens, it causes a brief change in the membrane potential. So it's a small change for a brief time. Okay, so I think you can see in the diagram here, let me grab my little pen. You can see here are my neurotransmitters. They are binding to a ion channel here, which is opening the channel. And in this case, some of these ions are moving through. I can't tell which direction they're moving. Okay. All right, so those postsynaptic potentials, those small, brief changes in the membrane potential, changes in the voltage due to ion movement, can be either excitatory or inhibitory. So we call it excitatory, or an EPSP, if it moves the membrane potential closer to zero. We call it a depolarization because then there's less difference between the inside and the outside of the cell. A membrane potential of 0 would be no difference. So when we're more negative, that's hyperpolarization. When we're less negative, that's depolarization. We're closer to equality, so to speak. So if you move the membrane potential from -70 to less than that, right? So to like -65, that's called a depolarization because now it's less different than the charge outside of the cell. Those are excitatory PSPs. Inhibitory PSPs cause ion movement that moves the membrane potential in the opposite direction, makes it more polarized, more negative, and this actually makes it less likely that we will send an action potential. So the ePSPs are the ones that are saying, do it, send a signal. And the iPSPs are the ones that are saying, no, don't send a signal. So we have these two different types of signals that are coming into the neuron. Now, these signals, of course, can add up. So you can have what's referred to as spatial summation. So these signals are coming in at different spaces, but at the same time. Or you can have temporal summation. So summation over time. Temporal means of time. So they're happening one right after the other. Now, each one of these is brief, but it's happening so that some are still at play before the other one wears off. So it's kind of like if somebody's on a swing and you push them, and then before they start swinging back, you push them a little bit more and push them a little bit more. You can get them to go a little bit higher. So that would be temporal summation. Here is a diagram from the Hedges textbook that I kind of liked. So they're showing us a postsynaptic neuron right here in the middle. And then these are axons coming from other neurons. So we've got input 1, input 2, input 3, and input 4. So axons from other neurons that are creating synapses with our postsynaptic neuron. So what they're showing us here first is just that axon number 1, that input number 1 is giving us a single excitatory stimulus. And so what that's doing is it's moving our membrane potential less negative. It's causing a depolarization, but it's brief. And so then we go back to where we were before. Now they're showing us temporal summation. So this is where before it wears off, you hit it with another one and another one and another one. It's from the same place here. It's still input 1. It's still the same axon. It's just firing its signals and releasing its neurotransmitter quickly. And what happens is because each subsequent one, where did my cursor go, is coming before the other one wears off, right? It builds, builds, and builds, and we see a greater depolarization occurring. This particular one is probably enough to reach threshold. Spatial summation is different. So now they're saying, okay, all 4 of these presynaptic neurons, all 4 of these different inputs are sending a signal pretty much all at once. There's a slight difference in the time there, but pretty much all at once. And so again, we're getting overall a strong signal. It's adding up. We call that summation, just like doing sums is adding things. So summation is when these signals add up. This is just the same exact graphic without animation so that when you refer back to the PowerPoint, you can actually see it. Now, of course, some of those PSPs are excitatory and some are inhibitory. So again, from the Hedges textbook, we're seeing the first one is what we just saw before with just an excitatory one, just a single one, not enough to reach threshold. And then over here, we're getting both an excitatory and an inhibitory. These are coming from 2 different inputs. So input 1 is giving us an excitatory PSP and input 3 is giving us an inhibitory. And so overall, we have no change in our membrane potential, maybe a little bit of one, but nothing. So nothing's really happening. Okay, so for most neurons, the action potential threshold is negative 55 millivolts. What does that mean? It means that if all of those voltage changes from the ePSPs and the iPSPs summate either through spatial summation or temporal summation to create a net change in the voltage to negative 55, or less positive, less negative than that, then the neuron is going to fire an action potential. So what you can see down here is in blue, they're saying subthreshold depolarization. So there was enough, I'm going to switch to a highlighter. There is enough to kind of get a little less negative, but it didn't reach threshold, so then we just kind of go back to where we were before. No big deal, nothing happens. But then in green, we see we got enough summation of ePSPs, excitatory, depolarizing PSPs to take us to 55. And if you get to negative 55, then shabang, you do an action potential. And we're going to talk about that in 3C. So the answer to how does the axon hillock decide is that it basically looks at the net total of all of these incoming signals and the net voltage changes that occur. If you get up to negative 55 millivolts, you're going to send an action potential. And if you don't, you're not. Okay, so in summary, neurons are constantly receiving signals from other neurons, which are going to trigger voltage changes across the membrane because ions are going to move, called postsynaptic potentials. That process is where a neurotransmitter binds to a ligand-gated ion channel. It opens, ions can move according to their electrochemical gradient. Depending on whether they're positive ions or negative ions and which direction then they move, that's going to change voltage potential. If it makes the cell less negative, is depolarizing, that's excitatory because it gets us closer to minus 55. If it is hyperpolarizing, makes it more negative, that's inhibitory because it takes us farther away from negative 55. If we reach negative 55, we're going to go ahead and send an action potential, and that's what we're going to talk about in 3C. See you then.